Good morning, everyone, and happy Wednesday. It's 10 a.m. here in Montana, so we're going to go ahead and get started. I'm Cheryl Rogers, and I'm the Director of Marketing here at Golden Helix. Our webcast today is presented by our CEO and President, Dr. Andreas Scherer. Today, Dr. Scherer will discuss best practices surrounding cancer testing pipelines. Andreas, the honors are yours.
Cheryl, thank you very much for the kind introduction. Good morning, everybody. My name is Andreas Scherer. Today's topic is cancer gene panels. And before we dive into the subject, I would like to spend a few slides on talking about Golden Helix for those of you who join us for the first time. We're in the business of providing analytics software for researchers and clinicians for actually quite some time. We're in business since 1998 and have since then accumulated really a huge number of users. We have thousands of users across our three software platforms and hundreds of organizations on a global basis. A few tidbits that are important to know about us. One of our major outside investors is GlaxoSmithKline. They invested in us very early on and have been with us ever since. And the other tidbit that we're very proud of, we were referenced last year by Gartner, one of the world's leading market research companies in their hype cycle for life science. So we're in this space, one of the companies that they think people should have an eye on. This slide here gives you an overview of who we are doing business with, some of the names of the organizations that have been with us over the years, and there's a very active number of publications going on. We're approaching about 900 citations now, and that number actually grows every month. Every month we learn about more publications that are coming out where we were used as the analytics engine. So the topic is cancer, and one of my favorite quotes is this one here: "Cancer is a disease of the genome." This phrase was coined over 100 years ago by a German cytologist called Theodor Boveri, and he studied sea urchins of all species and came to very profound insights in his work. And it actually took a while for his insights to be covered in the English-speaking literature. It needed to be translated, so most of his findings were really recognized decades after his death. But this phrase, "cancer is a disease of the genome," is so profound because it really is the premise for everything we do in this space. It's the reason why we're looking for new genes that are involved in cancer. It's the reason why we're looking to understand pathways where we develop new therapeutic options that can treat cancer, and of course we're looking for the reason why we're looking for diagnostic capabilities, and it's pretty much the reason why we have this call here today. Just in case you have questions during this webcast, feel free to type in your question in this little panel here on the right, and at the end of the webcast we'll take a few minutes to go through them and try to our best to answer those in the available time. So I want to start more on the 50,000 foot level and give you a picture of what really cancer means today for us on a global basis. So this is the incidence distribution of cancer on a global basis. And you really can see in this picture that cancer is a disease of the developed countries. Strong occurrence here in North America, West Europe, Australia, and then some parts of Asia. And this is pretty much in those countries we tend to get older. We also make certain lifestyle choices. So that's pretty much what we're dealing with right now. We had in 2012 about 12.1 million new incidents, and I'd like to add to that this figure, which basically tells us it's really a serious disease. We had in the same year over 8 million deaths, and this is like the distribution of the cancers that led to death in that year. So really the upshot is that cancer, as it was in 2012 and still true today, is a serious disease. We lose loved ones, family members, friends, patients, and it's also a disease where we spend on a global basis quite a bit of resources. So we invested in 2010 over $1.1 trillion in trying to treat cancer, and there's really a question that comes up in all of this, I think the best way to look at this is in the US, for instance, we had quite an impact on heart-related diseases, which is still the leading cause of death over the last 10, 15 years. But if you compare the same statistics on cancer, it actually didn't happen. We didn't move the needle, and that is very astonishing, given all that research and all the resources that are going into this space. So how can we change this, and how can we move the needle, and what other paradigms are required? Let's look at lung cancer as an example. So lung cancer today comes in two major subtypes. There's the small cell lung cancer and the non-small cell lung cancer. And the latter, if it's being observed early, if you find a patient with non-small cell lung cancer early in the process, we can treat it with conventional surgery, and there's a good chance of survival. But in all other cases, the best option for many years has been chemotherapy. And the problem with chemotherapy is that really the response rate of the therapy is very low. It's up to 20%, but that means only 1 out of 5 patients really see some impact of that therapy. Now, in the last few years, there have been a number of targeted therapies developed that are now on the market. And for lung cancer, for instance, if there is a gene mutated in this particular cancer called anaplastic lymphoma kinase, or short ALK, then there are treatment options available. Those two here, crizotinib and ceritinib, which can improve the outcome quite significantly. So the tumor response rates here are over 60%, which is of course dramatically different compared to chemotherapy. And I just want to give you a very visual picture of what this impact could look like. This is a patient who got treated, had basically a severe case of lung cancer, got treated with ceritinib, and after 3.5 weeks, we see this dramatic improvement in treating this disease, and so you see that how the cancer here has receded. Now, why is this not exploding? Why is this not happening everywhere? And I think this paper that came out last year from Kelly and Alter in the American Society of Clinical Oncology is, I think, very helpful to understand where the remaining issues are. So they did a very comprehensive study, phase 3 study, that compared crizotinib with standard chemotherapy. And from a medical perspective, I think there is really no question that the impact of these new therapies is much better. So the median progression-free survival is over 7.7 months versus 3 in chemo. The tumor response rate is 65% in this study versus 20% in chemo. But most importantly, the therapy itself is much more tolerable. In chemo, the patients have to take days out of their schedule every so many weeks, and it's very hard on the body. People lose hair. It's a very traumatic experience. Not so much with these new treatments. We're basically taking 2 pills a day, and that's about it. You don't see any of these side effects. So it's not a surprise that 65% of the patients that had an option during this trial that moved over to crizotinib, given the chance. The paper goes very far in explaining what the real remaining issues are. So for once, these drugs are still very expensive. So 60 pills, which is basically 1 month's worth of treatment, are about $11,000, and there are a number of payers in other countries who basically already said a priori we can't afford this, and so we will only on an exceptional basis approve those treatments. So cost is from a drug perspective an issue, and then drug companies are here now asked to help. But the second issue is about the diagnostics of those. So as these drugs get approved, there's typically a companion diagnostic kit that allows us to test for, in this case, the ALK mutation. And we know a priori that only 3 out of 100 patients will have it. 3 out of 100 lung cancer patients will have it. So we'll spend easily $25,000 just on kits to test for it, and then we know out of those we have 3 patients that then can go on to a very expensive therapy. So that's a remaining issue, and quite frankly, the actual testing costs will be actually multiples of that 25,000, but we still have to pay for administering the kits and doing everything that is required to basically complete the whole process. So that's one issue. The other issue is that these companion diagnostic kits actually also create a workflow issue for the treating oncologist. And I was just a few weeks ago at a conference in San Diego talking to practitioners, and we talked about this over dinner quite a bit. So the problem is there's a patient sitting across your desk, and you know it's lung cancer, and now you have this companion diagnostic kit as a tool. So what you will have to do is you have to go and test for ALK, and then you test for a few others until you may or may not get lucky. And so the first issue is that we're running very quickly out of sample tissue. So we have to then go back and redo the biopsy to obtain more DNA. That's one issue. And then the second issue is that we run out of time potentially. Well, you can imagine once you have a patient with the lung cancer disease that we really want to get going on treatment, and so time is precious. But then there is a very fundamental problem with it that we simply can't solve in this paradigm, and that is there are right now maybe 20 or so drugs approved, but there are hundreds of clinical trials going on with different experimental treatment options, and the reality of today's world as a treating oncologist is that you're equally interested in finding approved treatment options as you're trying to find a clinical trial that potentially can help your patient, and you will not get anywhere with these testing kits. They will not be helpful whatsoever. So we're looking for a better, more efficient paradigm. And I think that's where cancer gene panels come into play, because the thought process is that we're taking a sample once, run it through a sequencer, conduct the bioinformatics, and we're really testing in this process not one against one potential gene candidate. We're testing all the usual suspects that are involved in cancer, where we know there are treatment options available, and the output of this process then is being used for diagnostic purposes. We're creating a treatment plan. Very often we hear that there is a review step. There's a committee that takes a look at this because still it's new technology and it's a new paradigm. And then from there's a final report and then patient communication. And so even though this sounds a little bit more complicated than just applying a simple testing kit, it's actually something that we can do within a 24-hour window. So this whole process could be completed very fast, and with that we would have a very comprehensive understanding of the situation and of the specifics of that tumor. Let me give you an example of one of those panels. I'm using here the example of the Ion AmpliSeq cancer hotspot panel from Ion Torrent, but this is really just an example, and I don't want to endorse this product. There are other products available from other vendors. Illumina has one, Raindance, and others. So this is just really serving here as an example. And what we're doing here is we're just taking, I think, a modest amount of DNA, 10 nanograms, and we use this panel as a tool, essentially, to look at very targeted genes that are involved in cancer, and where we know their treatment options are available. As you see here, for instance, ALK is one of the genes that this is part of the test. What really this is the majority of this is chemistry, and we're able to zero in on hotspots and regions that are relevant in cancer, and these kits amplify those regions. So it means we have a higher coverage later on during sequencing. The interesting thing is that this whole part can be dealt with in about 12 hours. So from sample prep through running through the sequencer, it takes about 12 hours, and then from there on we just look at data. And I would like to spend a little bit of time to give you an overview of what on the bioinformatics side of this issue we're dealing with. So this is an overview of of a somatic variant discovery pipeline. So this is it's a little bit more of a general case. So we would sequence the tumor, sequence the normal, align those against the reference genome, and then in that case here, we would look for somatic SNVs and indels, but we also would look for structural variations where the chunks of the DNA have been moved to another part of the genome, and we would look for copy number alterations. After that, there are certain QC steps, quality control steps, and then there is an annotation step that basically enriches that data set with everything we know about those variants at that stage. In a testing lab where we just apply a straight up gene panel, we would actually just look at the tumor itself, take that sequence, align it against the reference, and then focus primarily on SNVs and indels, and then apply the QC and the annotation. So that's the difference between a pure testing pipeline versus a discovery pipeline. It would be more appropriate in a research setting. We have a wide range of people coming to these webcasts. For some of you, this is standard bread and butter, and for others, I would like to just point out that really the alignment portion of this is really solving like a puzzle. The human genome is 50% of it is at least highly repetitive, so solving this alignment problem is not trivial. The exome is a little bit more differentiated, so it's a little bit easier to match the strands that are the reads that are output of the sequencer to the correct region. By and large, this problem is from a computer science perspective, it's NP complete. That means we are not able to come up with an optimal solution efficiently. So that's the reason why we see in this field a plethora of different algorithms. I think the first were invented in the 1970s, and but we see ever since a constant progression and evolution of new algorithms that are solving this type of issue. How this looks really, and from a data perspective. So the output of the sequence is what's called a FASTQ file. You see here on the left side the reads as an example. In reality, these reads are between 50 and 250 base pairs long, and then we try to align those against the reference in a second step. This is being stored as a what we call a BAM file, and then from there we can identify differences in these aligned sequences against the reference, and you see those here marked in green and red. That information then is captured in a VCF file, which is the input of what we are doing here at what we're using here at Golden Helix. You could go ahead and visually see these variants if you would go back and take a look at the BAM files themselves. And sometimes after everything is said and done and the whole process of the secondary and tertiary analysis has been completed, some of our clients actually go back and do this just to confirm that the variants that have been identified are not just an artifact of the process, but are real. So you can really see this here in a comparison where you see on the upper half the normal sequences, the strands of the normal cell tissue, and then on the lower part you see the tumor sample. And there are some reads that are showing a variance. It's certainly suspicious. Of course, this will be a long day in a lab if you would do this by. So we employ typically algorithms to detect those, and an obvious choice if you are on an ion torrent platforms, which a lot of our clients are, is to use their built-in capabilities. So they have a proprietary alignment algorithm called TMAP, and then they have a variant caller. It's really a set of 3 different algorithms that then create an VCF file as an output. On this subject, there is a very extensive webcast on how the secondary analysis looks like from Gabe Rudy last year. So welcome to take a look at this at your convenience. It goes a little bit more in depth in what the available algorithms are and explains them at least on a high level. So once we're past the production of a VCF file, it's really about finding out what the quality of the data is. You see here a screenshot of Varsik, which is our product for this space, and so coming in, we have in this particular example 1,708 variants identified, and we apply certain filters. One important one is to ensure that the read depth along those variants is really sufficient. So we're asking here for a read depth of 1,500, and that whittles out about 181 variants that are not sufficiently covered. And then the second filter, which is even more stringent, asked for a minor allele frequency between 1% and 30%, and that just is a good number that is being used as a best practice to zero in on what are potentially suspicious and somatic variations. And as a result, then we have 5 left, and then of those 5 we can ask the question, are they known, are they covered in cosmic, yes or no? And if so, the ones that are uncovered, they are displayed here on the upper right, and there are further drill down capabilities that are available. So we can very quickly, this whole process up to the VCF file probably took us 16 hours. This type of analysis can be done in Varsik just within seconds or at least minutes. What then will happen is we would take a look at annotation sources to learn more about these possibly somatic variations, and we would go in Cosmic, which is actually shipped with so that this data source is actually shipped with Varsik, so it's so we have this as an annotation source already built in. But there, of course, there are other sources that our clients use, such as the Cancer Genome Atlas or clinicaltrials.gov, where you can learn about ongoing trials in this space, or My Cancer Genome. What we wanted to do, though, at this point is to give you a little bit more of an in-depth understanding how Varsik can be used in this context. And I have here today with me Dr. Bryce Christensen, who is our chief scientist, and he has prepared a few examples that he's ready to show now. Bryce, let me hand it over to you.
All right. Thank you, Andreas, for the introduction and for all of the great background information on cancer gene panels. What I would like to do here over the next several minutes is give our audience a general overview and introduction to the Varsik software. We'll look a little bit at how it works and some of the options that are available for data analysis within Varsik. We saw a really nice screenshot there a few moments ago in Andreas's slides. Now we're looking at the live software running on my desktop. What you find when you run Varsik is that it is an environment that allows you to read in variant calls from a VCF file and proceed to apply annotations and decision rules based on those annotations so that you can select particular types of variants for analysis. So what we see here in this particular data showing on my screen is initially you have some basic data that comes from a VCF file. So I can see here the mapping coordinates for a set of variants, the genotype calls that have been made, together with some technical data that would come from a VCF file, such as the overall coverage depth or the number of non-reference alleles that are observed in the coverage statistics. Now, beyond that, we have a number of annotations that have been generated and applied by the Varsik software. So we can see, for example, what gene each variant is located in, what type of variant is it in terms of being synonymous, non-synonymous in a UTR or a splice site, for example. And the nice thing in Varsik is that it gives you extra information. So clicking on any one of these columns will give some detailed information about how that data is generated, together with either a distribution for a numeric variable, or in this case we see the frequency counts for this categorical variable. And as we move across to the right, we see here a number of other annotations have been generated. There's extensive flexibility to use different public databases to bring in your own proprietary databases for analyzing your data. Now, once you have annotations applied in this manner, it becomes a fairly straightforward process to set up what we call a filter chain or a set of decision rules to analyze the data with. Now, in this case, I also have several samples that have been imported into this one project. So I can move from sample to sample and see how the data changes in each sample. And here on the left, I have my decision rules set up. And this particular workflow has been organized in two different branches, where in a diagnostic lab, for example, you might want to start by looking at known mutation hotspots. So here we've taken a total of 1,257 input variants for this particular sample that have some evidence of a non-reference base. So I can see that entire list of 1,257 by clicking on it. But I can also see that at known mutation hotspots, with some minimum coverage depth and a minimum number of alternate alleles present, I have just 3 variants. I can click here, see what they are, something in BRAF, KRAS, TP53. These are all important cancer genes. Now there's also a secondary workflow that's been set up for this particular example, where we have mutations occurring away from those known hotspots, where we're applying similar rules, confirming some minimum amount of coverage, a certain presence of alternate alleles there. We have thresholds set for this alternate allele frequency, or the allelic fraction, in terms of what proportion of reads are for the non-reference base. There's a certain range where we generally expect to find somatic mutations. Of course, it's possible to be outside this range, but this is where we normally start looking. And we see in this particular sample, there are 295 potential somatic mutations being identified. I can see all of them here. But then to whittle it down a little bit, we compare it with dbSNP and see how many of these are common in germline tissue and therefore might just be false positives. Since we don't have matched normal for this gene panel data, we have to use what's available. There are 83 of those that are rare in germline tissue. Additionally, just 12 of them are in cosmic. So if we want to learn what's already known about some of these, we can pull up that list. And beyond that, we added one final filter for this particular template to see how many of them are loss of function or missense mutations. Now, one of the really nice things in Varsik is that we're able to interact with external data sources. If I make my window a little bit bigger here and just select one of these variants, maybe if we'll come back and look at KRAS. When I click that, you'll notice that the detail pane updates, and I can see a lot of additional information regarding each one of these mutations, including a number of external hyperlinks. Now, this happens to be a very common variant that has been reported many times. I have all of these different PubMed references that I can select, but all of them are active hyperlinks. I can pull up abstracts. I can pull up records from Cosmic database. I can pull up records from NCBI and other resources. But if there is knowledge about variants available, we try to make it accessible from inside the software here so that you can learn about it. Now, in this case, where we have a KRAS mutation, that might indicate reduced sensitivity to anti-EGFR therapies and is therefore clinically relevant and important to know about. So this is one way to use Varsik. You can import data, you can take multiple samples, you can see how the data changes from sample to sample, generate individual reports for export. But Andreas also pointed out there are research workflows as well where you might take different approaches to the data. So I have a second instance of the software open here to look at, just to give everybody a flavor of how Varsik works with exome scale data. So what I have on the screen now is a matched tumor normal pair. This is a gastric cancer pair that's publicly available through SRA. But after aligning the short reads and making variant calls using a somatic variant caller, we have in this case nearly 500,000 potential variant sites, 455,000 through the exome. And you'll notice that Varsik is very capable of handling this large-scale data. It smoothly scrolls through very large tables, in this case 455,000 rows. And as we examine the data more closely, we see we have data related to the normal sample, so called genotype, read depth, some somatic scoring options coming from the variant caller, together with this allele frequency or the allelic fraction for that alternate allele, and similar data from the tumor. And again, we're able to set up decision rules based on any of these variables, whether they're from the VCF file, as we see here, or if they're coming from external annotations like we see out to the right. So in this case, we set up a fairly simple template, initially identifying the 386,000 variants that are flagged as potential somatic changes by the variant caller, which was very sensitive. Then confirming some minimum amount of coverage individually in the tumor and normal samples, so we have some level of comfort with what's being detected. And from there, moving on, looking at the allelic fraction in the normal and the tumor. So in this case, we used slightly different numbers. We removed anything where the allelic fraction in the normal was more than 1%, and focused on things in the tumor between 5 and 40%. We see 196 potential somatic mutations there, and just updated the list of those 196, and further added another filter based on simply, is it expected to be loss of function or missense? So here we see this final list of 13 that are all either missense variants or stop gain variants. And so you see that Varsik makes it quite simple to start from a very large list of variants, reduce to the variants that fit a particular profile. Of course, you can change your filters at any step along the way. One thing you may want to do is visualize some of the raw data to really take a closer look at this. And so right now we have this first variant highlighted that's in the ARID1A gene. Varsik has an integrated genome browser that's based on our free Genome Browse product. And here we're looking at the BAM files, or the alignment data, for both the normal sample at the top and the tumor sample at the bottom. Let's start by looking at the tumor sample at this particular locus. If we expand this out and zoom in a little bit, you'll see that we're able to view the individual short reads that are aligned to create the sequence in next-gen sequencing, and see all of the base pair level data here. Now, you'll notice that anything that doesn't match the reference is highlighted in a different color, and in this case it's a T, so that color is blue. And we can see here the relative proportions at the top, so it's covering maybe a quarter of the total depth. But we can get detailed data simply by clicking. Everything is always clickable in Varsik. In this case, the details are going into this detail window. I'll move it over here where we can see it a little bit better. But it's telling us there are 98 total reads at that coordinate. 21 of them are for that mismatched base. Now, if we look at the normal sample, let's just highlight that area so that we can see it better. Come up to the normal. Here we can see 74 reads. They're all for the reference allele, so there's no evidence of this mutation in the normal sample. Again, we can zoom in and take a closer look here, but we know that there's not going to be anything to find. So this is a great example of what you might look for if you're searching for somatic mutations. So hopefully then you've been able to see a little bit about the utility of the Varsik interface here, the flexibility, the capability for annotating variants, for filtering variants, creating reusable templates as well. So all of these rules that we've set up for filtering in both of the two projects we've looked at can be applied in the future to new data sets to really automate your processes. And with that, Andreas, I think we'll give control back to you.
Yes, Bryce, thank you very much. It was a very good presentation. I have just a few topics that I would like to touch on. We just 48 hours ago, we just finished a deal with a company called MedGenome. And it's a very recent development. We didn't even have a chance to put out a press release or blog post or any of this. So really people here on this call are the first to hear about this. As you can see, it's one topic or one issue to identify the variants and make sure that they're real and that they're relevant. And then there is the next step where we basically have to assemble a report, and there are these different data repositories and resources we can draw on. What they have done is they've created a database called OncoMD that basically includes all the information that there is to know about somatic variations and any cancer data. So you can get annotation information, information about genes, drug information on and off-label use, response rate, and active clinical trials that are relevant in this context. So what this actually does is it speeds up the time it takes to build a final report, and really condenses the time to go from sample prep to patient communication. So that's a very big step here for us. We're going to complete the integration into Varsik soon, so that this information is really available at your fingertips. The second point I would like to make, and let me switch on here the demo, is that we started with Varsik really in the fall, in October last year. We had a webcast like this one to introduce it to the public. And the interesting thing was, while we were at the webcast, there were people calling in and wanted to buy the product. So we had a very, very better than expected start with Varsik. We just launched the 1.11 release this Monday, and that has a number of features that are clinically relevant. So there is for those who are doing or conducting germline looking at analysis of germline mutations. There's a gene ranking capability now built in called FORank, which allows you to faster zero in on relevant variants. We are supporting a custom variant database, so as you're progressing in your clinical work and you find interesting or important variants, you can store them in this database, document them, and then annotate this against this database in all of your future sample runs. There is a log file capability that tracks all the activities that a user does with a given sample, and that is from a compliance perspective an important feature. And then we have some slew of other capabilities that we added in this release. It's very powerful and an important release that we just launched. The product was presented at ASAG, and then just at the last TriCon, we showed it there also in one of our presentations. So, and we have we were very lucky with a lot of clients jumping early on this product. We had a few key announcements that were covered in press releases. So, for instance, North Shore Next-Gen Sequencing Lab is one of our clients, and then a big one is Prevention Genetics and their testing lab. And this story was actually covered by Genome Web. So we're feeling now that this introductory period is coming to an end. What we have done is we gave strong incentives from a discount perspective to our clients. We also gave those clients a 15-month license as opposed to a 12-month license. So that process is now coming to an end. I will say, though, is that we continue to be at a very attractive price point, and we also stay away from any per sample pricing. This is, I think, one of the things that some of our competitors do, and it seems to annoy our clients, because just from a clinical perspective, sometimes you have to rerun a sample not only once but twice or more often until you get the results you want, and so it really creates a gray area. Is this something that needs to be then charged extra, or is it part of the initial run or so? So we stay away from this issue. So we'll continue, of course, to develop the platform. There will be that we have a very, I think, aggressive development pipeline for the remainder of the year. And then, in addition to that, we're continuing to round up the product and the value proposition around it with partnership like the one that I just mentioned with MedGenome. So this is a very active space, and we do these webinars to inform our user base, but also I think the whole community. We have a lot of people that are not our users that come here to these events. Equally interesting might be for you the fact that there is a ebook out there with the title Genetic Testing for Cancer. I think this came out late January of this year. If you follow the link on the bottom, you get to the download page, or you simply go to goldenhelix.com and look for the ebook section, and there are a number of choices, and I think this is the last addition to the ebook section. I would like to finish this webcast up with an outlook on where we stand with this paradigm. I think it's clear by now that gene panels are a much more superior, more efficient diagnostic option than these companion diagnostic kits that are right now the standard that come with these very specialized targeted treatment options. But the question is, where do we go with this? And in this context, I think this publication from Lawrence et al. In Nature last year is very interesting. So what they did is they looked at the literature and then the studies that were that we know of at that time, and this was covered about 5,000 tumor normal samples across 21 of the major tumor types. And the paper is very good in outlining so how is this field progressing and what can we expect. So it's fair to say that we probably have discovered those genes that are mutated 20% of the time or more often. So it's very unlikely that we find another gene that is mutated at that frequency. But there are a number of other buckets. There's this category 10 to 20% mutation frequency, 5 to 10, 3 to 5 to 3, or less than 2. And what is true for all of these categories is that we still continue to learn more about genes that are involved in cancer in any of these categories. In some, it's starting to slow down, like the yellow one as you see here, but by and large, we'll still increase our knowledge. And what that means is we will have more opportunities to identify pathways that then can be treated with new treatment options. So by and large, what this means is that gene panels will be more complex on one hand, but on the other hand, they are probably for the time being a very, very attractive option in the clinical realm to answer a very simple question. Can we solve this? Is this patient treatable? Yes or no? So with that, I would like to conclude this webinar and hand it over to Cheryl for a few announcements.
Thank you, Andreas. I see the questions rolling in, and we are going to go ahead and answer those in just a moment. If you haven't had a chance to type yours in yet, please go ahead and do so. If we don't have time to answer them, we will be in touch later. I should have the webcast recording and slides up tomorrow. If not tomorrow, then Friday. Our next scheduled webcast is coming up quickly on March 25th, and that will be presented by Dr. Jeffrey Moore of the University of Illinois Urbana-Champaign. Dr. Moore recently introduced a new curriculum integrating genetics into organic history education, and it should be a very interesting webcast. I should have the invitations starting tomorrow. With that, Andreas, I'm going to hand it back to you for questions.
Okay, good. So we have a few that are very interesting. One is I think going back to this last comment on gene panels and why are we not simply doing whole exomes going forward. I think really and this is a valid question. We obviously discuss this in our field quite a bit. The reality is, as of today, gene panels are, they're answering really a very targeted question. So we're looking at clinically actionable genes. Right now it's 50 or around that, versus as opposed to 20,000 in a whole exome. Coverage is an issue where we want to have a coverage of 1,000x or more versus for exomes we get right now as a standard 200x. But really the last issue is that and why the clinician probably would like to have something like a gene panel as a preferred choice initially is it really answers the question, do we know it? Do we know this cancer? Can we treat it? And if yes, then the work of the clinician continues. If not, it becomes more of a researchy type of activity. And there's also, of course, the issue of incidental findings. So if you have a whole exome analysis and you look at everything from across the board, and you might oversee an issue that has nothing to do with the cancer diagnostic that you're dealing with right now, then it's unclear how this could be seen or could be handled. So it's from a clinician, from an oncology perspective, it's a very attractive paradigm just to look at this as a very targeted test. Okay, but very good question, and we might change our answer in a few years if we have a better approach or a better way to deal with this. There's one question, maybe Bryce is for you. Also, do we have the flexibility for choosing our own annotation program with the Varsik software?
So Varsik, of course, has all of its own built-in annotation capabilities. Everything that I showed today was using Varsik's own annotation engine, which we have very carefully constructed over time. If you do have your own preferred annotation source that you like to use, yeah, we can work with that in different ways. Number one, if you have an annotation database that you would like to access from within the Varsik software, we can configure it to read from different data sources, different databases. And if you already have extensive annotation data contained within your VCF file, as is often the case, there is nothing that prevents you from building filtering workflows around those pre-existing annotations within your VCF file. So yeah, a lot of flexibility in terms of how annotations are generated and how they're used.
Yes, okay, great. There were some questions about the partnerships that I talked about, so I'll just try to summarize them. So timing on the Met genome integration, it's a little bit of a TBD. We hope that it is actually very fast, so hopefully with the next release. Worst case, so which will be in another month or so. Worst case, it will take us 1 month longer than that, but that's roughly the time frame that we're looking at. We're going to have additional communication and communication about the subject as we're getting closer to that to occur. And then apparently I didn't cover the Fluxion Bio partnership that we entered. That was something that we announced at TriCon a couple of weeks ago in San Francisco. The topic right now is that we have that there is a increasing focus on leveraging circulating DNA for not only diagnostic purposes, but also for the ongoing monitoring of a tumor. And they have developed a very neat solution for this, and we're their diagnostics platform. So it's actually here reverse. They are reselling our software, Varsik in particular, to their clients. So maybe, hopefully, that covers that. There's one question that I think that was covered by you, Bryce, but I would just want to make sure that we have this explicitly clarified. So the question is, if one does have a normal tissue, VCF, can Varsik filter the germline variants out? I think you covered it, but maybe it's worth to talk about.
Yes, and I think that question came in before we got to that part of the demonstration, yeah. But absolutely you can compare and contrast between different sample data. If you have tumor and normal data and you want to remove anything observed in the normal from the list of tumor variants, that is easily done within the software.
Yes, okay. And then there were a few questions around pricing, Varsik, and of course OncoMD, and what to expect, and what the end of the introductory period means. Maybe so much. Maybe just let me tell you, we take our community outreach very far. So we have on these calls also competitors, and so we all love them. We all keep each other on their toes. But I would like to stay away from actual numbers. But maybe so much. There are discounts involved that are expiring, and there is this 15 months license that expires for Varsik. We're not doing this past March. If you have further questions, just feel free to contact us, send us a pricing request, or contact us as a reply of this email, and then one of our territory managers will be in touch with you to discuss with you in detail. With that, I'd like to conclude this webcast. Thank you very much for joining us this morning. And again, we have a few events. We have a few webcasts coming up. The next one in a couple of weeks on leveraging our product for teaching purposes. With that, thank you again, and hope to see you soon on one of our next events. Have a great day.